All right, these are your notes for your DNA section on Chapter 8 in your Blue Book, and let's get started. So, DNA, the initial discovery and function of DNA came through multiple steps, starting back in the 1800s. The first guy to kind of look at this molecule and talk about it was Miescher, and it was in 1869 he extracted DNA from his cell and discovered that it was rich in nitrogen and phosphorus. But he didn't know what it did.
Dan Kellett, please report to the Media Center. Dan Kellett, you're needed in the Media Center. Thank you.
Griffith tried to create a pneumonia vaccine from pneumonial bacteria and in mice, and he discovered this process called transformation. So here is a quick little picture of the mice experiments. He added non-virulent, so non-killer bacteria, does not cause pneumonia, to mice, and the mouse, of course, lived. Then he added virulent bacteria to the pneumonia, or virulent pneumonial bacteria to the mouse, and the mouse, of course, died. And then he added bacteria that was heat-killed. So this was previously virulent, but then he added heat to it and killed it, and then the mouse lived. He was like, okay, cool, we can kill bacteria with heat. We kind of knew this by now. But then he mixed this fresh, normal, not going to kill the mouse bacteria with the dead bacteria in a vial and allowed that to cook for a little while, not cook as in heat, but just do its thing. And then when he injected it into the mouse died. So he figured out that these non-virulent bacteria can pick up DNA from their environment, from these dead bacteria, and they can actually get that bacteria to be incorporated into their DNA, and it can change the properties of that bacteria, and that process is called transformation. You have Avery and McCartney discover that the transforming principle that Griffith figured out was only in DNA, and therefore it was a hereditary material, that it isn't in proteins, that same process and that concept isn't available in lipids, and it's not available in carbohydrates. It's just in this DNA material that's rich in nitrogen and phosphorus. Then Hershey and Chase did an experiment with a bacteriophage, and they looked at radioactive DNA. So here's Hershey and Chase's experiment. They looked at different parts of a bacteriophage, which is a type of bacteria or a type of virus that can only infect bacteria. So they labeled its coat, and then they labeled the DNA inside the coat in two different experiments. So in the coat, they noticed that after the bacteriophage injected its substance into the cell, the labeled substance was no longer there. They couldn't see it anymore, so they didn't find sulfur in the cells, which was the part that was labeled in the protein. But then after they labeled the DNA with radioactive material and it injected that into the cells, they found that DNA was in the cells afterwards, so phosphorus. So they learned that bacteriophages actually do their work by injecting DNA from the bacteria into the cell. So all of these scientists came up with different properties that a molecule must have in order to be considered a hereditary material. The first one is that hereditary information must be transferred along with the molecule. That's where we were talking about in Griffith's material or Griffith's experiment, where it not only picked up the molecule, but it changed what that bacteria could do. Number two, each cell of a given species should contain the same amount. So your skin cells and your muscle cells and your retina cells all contain the same amount of DNA. Number three, the molecule has to be exempt from change because it is the genetic bridge between generations. So you don't want this molecule just randomly changing on its own. In those cases, they're called mutations, and those aren't always a good thing. And number four, must be capable of encoding a huge amount of information to build a new individual. Remember, there are multiple tens of thousands of base pairs in a human, less than that in other organisms, but it still has to contain a wide amount of information in a very little amount of space. So let's talk about the structure of DNA. One nucleotide has a 5-carbon sugar, which is called deoxyribose. So here's my lovely little DNA molecules over here. I've got this little picture of this guy, and then I've got a much bigger picture. It's down here. So this blue part over here, this is my ribose. This is my ribose sugar. Whoop. Over here with the A, T, C, or G, my ribose would be part of this guy holding it together. These guys are kind of like my backbone, and then these here are my nitrogenous bases, where A and G are the pyrimidines, and T and C are the purines. Oh, I might have that backwards. I'll have to relook that up. I always mix them up when I'm giving notes, and then the kids are like, Mrs. Compton, you told us wrong. Sorry about that. So if you look over here, the sugar is attached to this part here, which is a phosphorus. And again, that would make up this guy part, so you can't really see it in this picture of the men, but that's what's going on there. So there are 3 blank groups. There are 3 phosphate groups. So phosphate attached to phosphate attached to phosphate. That's these blue circles over here. And then one of 4 nitrogen containing bases, which are adenine, thymine, guanine, and cytosine. So depending on which base you're looking at, you have A pairs with T, C pairs with G, and they will either make 2 bonds or 3 bonds in between them. C and G both make 3. These little dots are hydrogen bonds represented here and here. And then A bonding with T makes 2 hydrogen bonds, and you can also see that in this picture here. The DNA is a polymer that consists of 2 strands in a double helix, which is the Watson and Crick model based off of Rosalind Franklin. The sad story about dear old Rosalind Franklin is she did this X-ray of DNA. This is actually what you see. If you take an X-ray of DNA, and I'm not going to get behind the science of why this works, but the structure can only occur in a double helix. And she was doing this and presented her work, and another person saw it and took it to, or Watson saw it, I think, and then took it to Crick, and then they built this model off of their research. So they were actually given the Nobel Prize, but by this time, poor old Rosalind had developed ovarian cancer and died, so you can only give the Nobel Prize to somebody if they're alive. You don't get to give one post-mortem. So this poor lady that developed this research and did all this work never got her prize for her work, but off the shoulders of a wonderful female giant there in the world of DNA came this Watson and Crick here, and this is an actual picture. It's been colorized, but this is a picture of their model of DNA, and you can see the 5-carbon sugar here, and they learned that the only way that the double helix could hold together was through weak hydrogen bonds in between bases. And so they were who took that double helix shape and kind of puzzled out how it worked based on the knowledge we had about the molecules that made it up. Hydrogen bonding between paired bases makes the rungs of the ladder and holds the strands together. A bonds with T, C bonds with G. That's Chargaff's rule. So dear old Chargaff is who discovered that in any organism, the number of A's and the number of T's are roughly equal, as are the number of C's and the number of G's. So they figured out that the cytosines have to be about the same number of guanines, and same for A and T, with the exception of a few base pairs because of always mutations and other extraneous examples in DNA. But for the most part, A will be the same as T in any given scenario. They also learned that DNA is anti-parallel, which is based on the arrangement of carbons with a 3 end and a 5 end. So if I were to draw a lovely carbon here with DNA, you would start numbering it at this point, or a sugar, my deoxyribose sugar, and start numbering my carbons. I'm sorry. There's carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon, oops, this is, shoot. I have messed this all up. I don't want you paying attention to any of that I just drew. Let's see if I can delete things. Delete, delete, delete. Sugars always have, and I know this, an oxygen in them. So let's try this again. So this is carbon number 1. This is carbon number 2, carbon number 3, carbon number 4, and carbon number 5 kind of makes up the chimney. And DNA runs from the 3 end to the 5' end, so it would run this way. And then the opposite strand of DNA would run the opposite way. So you would have your chimney piece here, and then the house runs this direction. So 1, 2, 3, 4, 5. And so this one would run in this direction. And then you would have your bonds in between them. going with the nitrogenous bases off of this number 1 molecule, and so that causes it to spin. But the anti-parallel means that you have one strand of DNA going upwards from 3 to 5 with your numbering of carbons on the sugar, and the other strand going downwards with the number of carbons on the sugar there. And the last one for this note segment is DNA sequence varies among species and among individuals. So that's what makes you unique, is the order of A's, T's, C's, and G's that your DNA comes in.